So in the last section, we talked about facilitated diffusion, where some things cannot go through osmosis, so they go through these protein channels to get in, moving from the areas of high concentration to areas of low concentration. So what happens when we want to go against that concentration gradient, when we want to go from low to high? That is where active transport comes into play. Active transport is going to require energy to transport small molecules across the cell membrane or through the cell membrane. Active transport is more effective than diffusion, but it costs the cells energy. And there's a couple of different ways that cells can do this active transport. So one of the ways that we don't have on here is pumps. So there are different kind of pumps that are going to help active transport. And one in particular, there's this sodium-potassium pump, and what it does is there is a molecule that basically attaches, this protein attaches to sodium and potassium ions and kind of helps them go against their concentration gradient. We're not really going to talk so much about this, but this idea that there are some pumps that are going to work a little bit differently than the next two ways that we're going to focus on. Two ways that for this chapter that we're really going to look at is endocytosis and exocytosis. So when you think of endo, think of entering. And exocytosis, going to be exit. So we're going to focus on how do things get into the cell and how do things exit the cell.
So this one, endocytosis. Endocytosis is the process of taking materials into the cell by the means of infolding or pockets in the cell membrane. So this little diagram here kind of walks us through. And remember, the endo, we want things entering the cell. There's got to be a way that we can get materials, molecules into the cell, even though the concentration doesn't match what diffusion should be.
So when we look at this diagram right here, this is endocytosis. So what happens is there is some type of solid particle, on the outside. And what happens is it gets close to the plasma membrane, and the membrane is actually going to surround it. And then it is able to kind of seal itself off and then come into the cell. So we get into this different vacuole where that solid particle now is. And it can look a couple of different ways. And again, the phagocytosis, where it's actually almost going to look like it's going out and eating it. The pinocytosis is where it's going to make an impression. And you have the receptor-mediated endocytosis. But what you need to know is the endocytosis is it's actually going to get things into the cell. And they form these little vesicles, vacuoles, getting those materials in the cell. So the other one that we're going to look at is the exocytosis. And again, exo, exit. So exocytosis, the membrane of the vacuole surrounding the material fuses with the cell membrane, forcing the contents out of the cell. So what happens is you have some type of vesicle that has materials inside it that they no longer want. And remember back to the cell parts, the organelles are membrane bound. So those membranes are going to be very similar to the cell membrane. So what's going to happen is this guy is going to move over here. It's going to attach to the plasma membrane, the cell membrane, and when it does that, because of its makeup, it's going to allow for these to be released. And we'll talk a little bit more in class about, again, the purpose of endo and exocytosis, but this ends the notes.